In this PowerPoint, we're going to look at how DNA holds the code for the instructions to build proteins and what these proteins may do. Remember, the DNA is a double helix and the rungs of the ladder are comprised of bases. The bases here, for example GCA, form a code. The code is read off by chemical machinery in your cell and translated into a strand of amino acids. Here GCA will mean the amino acid alanine is selected. AGA will mean the amino acid arginine is joined on. The bases GAT will mean asparagine or aspartic acid is joined up. And lastly, AAT, and this one I think is asparagine, is joined in position number 4. Now a protein is simply a long strand of amino acids. So the code in your bases defines the structure of the protein. What are proteins? Well, they're simply a long strand of amino acids, and based on the amino acids that are present determines the shape of that protein, and the shape of the protein defines what that protein does in the cell. If the sequence of amino acids changes, the function of that protein may change, or that function may be lost. The main groups of proteins are actually proteins that form something structural in your cells, such as muscle fibers. There are genes for the substances myosin and actin, and they're the muscle fibers that make up muscle bundles and muscle filaments. Other proteins just may make important molecules in your cell. This protein down here, showing the long strand of amino acids, is the protein that causes this colour in lizards. When these 3 amino acids here are exchanged for different ones, that protein can no longer produce the brown colour and the lizards are white. This is also the protein that makes hair colour, and a few changes in this amino acid produces red hair colour. The red hair colour mutation in humans is a comparatively recent mutation. Scientists think that red hair was first appeared in humans at about 15,000 years ago. The last most important type of protein in cells are proteins which are enzymes. Enzymes control the rate of chemical reactions in cells. They make cell chemistry possible, and that chemistry causes features to happen in organisms. This is an enzyme here which catalyzes the breakdown of sucrose into glucose and fructose. So it's specific for binding to this molecule of sucrose and causing that reaction to happen. So how does that sequence of A, G's, T's and C's hold the code? Well, there's your DNA, and your cells take a copy of that, called RNA, kind of like taking a photocopy, and it goes out into the cytoplasm where machinery in your cell reads off that code and as a result builds a specific chain of amino acids which builds a polypeptide, part of a protein, and ultimately a protein. In this copied molecule, U's or uracil are built in instead of T's, but you don't need to worry about that. So the DNA doesn't build a protein, it's like a blueprint. And the cell makes a copy of that sequence and leaves the nucleus, as we saw in the previous slide, to be read off. And the protein is made up according to that code. So if the sequence of the bases change, the sequence of the amino acids in the protein will change, and it may now not function as it should. So if a mistake was made during DNA replication and a C was placed there instead of an A, and the little proofreading enzymes that you've got in your cells didn't notice, the next time this is copied, you will have a G put there to bind to the C. So now instead of a TA pairing, you've got a GC pairing. And when the DNA is read off, a different amino acid will be put into that protein. As a result, that protein now may no longer be able to do what it did in the cell, or it may do something different. Blue eyes have their colour because of a single genetic mutation that occurred about 10,000 years ago. After studying 800 blue-eyed people, researchers identified a single base pair change in the human eye colour gene that all those people had and none of the brown-eyed people had. So that's the mutation that caused blue eye colour.